Let's go. What day?
You have to practice this. Okay, started. So very appropriately, the smell of citrus is in the air, right? Our speaker today is Christopher Vincent. He's at the Citrus Research and Education Center at the University of Florida, which is I'm blanking on the name, the city.
Lake Alfred.
Lake Alfred, yes. So, and so before that, he did his PhD at Florida as well. He's from Arkansas, so I just found this out about Christopher today, that he did a BA in journalism and Spanish, and then said, actually, maybe I'll move into horticulture. So that's sort of an interesting thing. And Christopher's focus has been on sort of the physiological basis of a number of things related to citrus, but specifically with HLB disease that he's going to talk about today. I think mostly talk about today. One plug, so this went out to a few of you, but we're tomorrow. So he does a lot of work in whole plant physiology, so very similar to stuff that we do in my lab and Scott's lab. And he has a paper that's a year or so old on gas exchange at very low conductance rates and some of the issues with that. So we're going to have a paper discussion tomorrow at Lavazza at 9 a.m. We'll find a spot. So any of you that are interested in whole plant ecophysiology, gas exchange, come along and we'll have a chat. And if you want the paper, just send me an email. I can send it to you. So, with that.
And so I don't need a mic for the
No.
Zoom people to hear. Yeah, great. Awesome. Well, thank y'all, and it's great turnout. So I hope that this presentation merits your attendance. Let's see, is this it's not working. That's all right. I'll do that.
Sometimes if you just
Maybe it's try it again. Let's see.
Yep, there you go.
Yeah, all right. So my lab is the tree ecophysiology lab at the Citrus Research and Education Center. And when people ask me what we do, what tree ecophysiologists do, what's that?
Sorry, I'm just trying to move this. No, oh, here we go. Okay.
Yeah. So when they ask me what we do, I say, depending on the mood, my mood that day, I say, oh, well, we sit around in the shade and we drink orange juice all day. Or I say, I'm the Lorax, I speak for the trees, and they're not very happy right now. So what we actually do is we do sort of whole plant or whole organ level physiology. We focus on sort of the intersection of carbon and water transport and photosynthesis. We have sort of an applied component and a basic component of almost everything that we do. And in terms of environmental factors, what we've tended to focus on is disease, which is almost entirely what I'll be talking about today, light distribution in the canopy, and then hurricanes, because we keep getting them, and so we need to know how they're affecting the trees. But I won't talk about the hurricanes today. So just as a little outline of what I want to talk about, I'll introduce what HLB is, and then I'll talk a little bit about source sink dynamics, sort of outside the context of the disease itself, and then we'll go into the tree and see kind of like how HLB is affecting trees in terms of source sink dynamics, and then I'll kind of zoom out and talk a little bit about potential applications of physiology to citrus production. And within that, we'll talk about like therapeutic delivery and environmental manipulation. Okay, so Florida has like a long cultural history associated with citrus production. What you're seeing here, these are prints of marketing from the 1920s, 30s, and 40s. And this is very common. You go into a bathroom in Central Florida, like a public restroom, and this is the wall art. Like in the diners and in the restrooms, everywhere you go, you see this kind of stuff. And it's all different. There was incredibly diverse art for everybody. Every farm had their own sort of marketing for the citrus that they were selling back then. So it was highly successful. It sent somewhere between 3 and 5 generations of Central Floridians to college, and successfully achieved a certain degree of economic stability. And then many challenges have occurred over that period of time, but HLB has been the biggest one. So I'll talk a little bit about what HLB is, but if you look, this is over time, starting in the 1960s, and you see kind of a general trend of progression. This decline in the 70s is related to freezes. You can see all those freezes that were occurring in the 70s and 80s. And then you see, you know, every once in a while a hurricane will have a big impact, but this green dashed line, that's when HLB was first detected in Florida. And by 2011-12, we had roughly 100% infection rate in the state. And by 100% infection rate, I mean like all the trees. So now if you go to Florida, if you see a citrus tree that's growing outside, it has this disease with almost certainty, right? And you can see what has happened as a result in terms of production, right? We're now at approximately 10% of the 2003-2004 harvest, which is pretty devastating, right? In 2003, there were 960,000 acres of citrus in Florida, and now there are roughly 200,000. So it's a pretty significant decline in acreage. Many people have left the business. It's been economically devastating, but the people who have gotten out of the business is even less than the decline in production, right? So the remaining acreage is less productive than what it was in 2003 and 4. So a big deal for people in the region and for the people who are growing citrus. So what is this disease? It's caused by Candidatus Liberibacter asiaticus. I'll say CLAS the rest of the time. I'll try to remember to say like the bacterium or something so you don't, you know, so that you don't have to remember all the acronyms. It is a phloem-limited bacterium that is thus far proven unculturable. So Cox postulates have not been confirmed. The infection is persistent. It lives in the phloem, distributes throughout the phloem of the tree, and there's no cure at a field scale for this. It's transmitted by an insect. And the common name is Asian citrusillid, that's Diaphorina citri. It reproduces on the new shoots, and that's the site of infection as well. So that reproductive cycle is where infection occurs primarily. Here you can see some of the symptoms. This is a healthy leaf. Here's what we call blotchy mottle symptoms. There's sort of asymmetric distribution of chlorosis, and that comes with hyperaccumulation of starch. So in this context where all of the trees that have been exposed are infected, a number of different practices have developed. One is there's a certain vector management in the sense of trying to prevent sufficient populations of the vector from like to slow transmission. Really doesn't work under our conditions. We can't get the vector populations low enough to slow transmission. But what we can do is we can put mesh bags over the trees, and that will prevent infection mostly up until the trees are so big we have to take those bags off to allow them to grow. So that works, it actually works quite well, benefits subsequent production to get the trees larger before they become infected. And then there's been a huge shift in terms of practices where the growers are much more focused on health promoting sort of horticultural practices, and that includes very conscious irrigation. You'll see here, like this soil here, the citrus soils in Florida are basically what y'all would probably call beach sand. It's roughly 95% sand. So very low water retention, very low nutrient holding capacity. So very conscious sort of what we call spoon feeding approach to irrigation and nutrient management actually helps the health of the trees, which you'll see why in a little bit. So there's been some success in using plant growth regulators to ameliorate some of the symptoms as well. I'll get into shade kind of at the end of my talk. And then something that really has turned things around a little bit, the last 2 years, we've stopped seeing declines in production. So that's unique over the past couple of years, and that's due to the introduction of the use of oxytetracycline and using trunk injection as delivery. So that's what you see there. That's a canister that's loaded and it's sort of under pressure, kind of like a water balloon. You drill a hole in the trunk of the tree, insert that plastic piece and open the valve in it, and the tree takes up that oxytetracycline, moves it around the canopy, and that suppresses the bacterium enough for the tree to recover some of its growth and yield. It's not a cure. It doesn't eradicate the disease from the tree. It just suppresses the bacterial titer and allows some recovery of health. So because of that, because it's not a cure, this has to be done, repeatedly, right? So about once a year, and as much as they're allowed to by the regulations, the growers are injecting their trees with oxytetracycline. Okay, so how does this disease work? As you might guess, with a phloem limited pathogen, it disrupts phloem function. That's not too surprising, right? And then we have a lot of questions, or we had a lot of questions in terms of how it disrupts phloem function. What's the pattern? So we had these questions, right? Does it reduce transport speeds? Does it reduce leaf export rates? Does it reduce photosynthesis through source sink attenuation? And I'll talk about what source sink attenuation is in a minute. And it does induce root starvation. That was one of the early observed symptoms. So really big decrease in root growth, and that leads to diminished water and nutrient uptake. And that's why those horticultural pieces, if they're well done, can be so beneficial. Is that you have reduced capacity for uptake, and so very careful supply, continuous supply can be really beneficial to the tree that's infected. But these questions that we had, the issue was that where that phloem function disruption occurred had a huge impact on the results of like what the answer to those other questions was. And answering those questions, we thought would allow us to sort of hypothesize like what you could do about it, right? So if we know where the disruption was occurring, then maybe we can hypothesize treatments that would be more effective, right? So I'm going to take a step back and talk a little bit about transport, carbohydrate transport and allocation, and how that relates to photosynthesis. And then we'll get into how those things are working in a healthy tree. And the reason why we had to get into how those were working in a healthy tree is that in order to understand dysfunction, we needed to look at function, but none of that work had been done either. So we just kept taking steps back until we end up with some relatively basic work, even though that's not exactly what we set out to do. So for those of you who don't think about phloem every day, the basic idea of phloem function is that you have a loading of osmolytes into the source tissue phloem. One of the main osmolytes would be like sucrose, and that induces uptake of water that leads to pressurization of the phloem, and then in the sink tissue you have unloading of those same osmolytes leading to efflux of water leading to lower pressure, and that pressure gradient drives mass flow from source to sink. It's a basic general concept. The main osmolyte being sucrose. If you have a decrease in transport, then what you have is a backup of all of those metabolites throughout the whole system back into the mesophyll. And so you have accumulation of sucrose, you have accumulation then of starch, and that disrupts all sorts of things in the mesophyll. And so plants generally have processes to sort of avoid problems with this, which is that this source sink attenuation hypothesis is that when you have accumulation of carbohydrates in the source tissues, you have downregulation of carbon fixation because you don't need to invest the nitrogen in producing lots and lots of rubisco if you're not going to use it, or if it's not going to be beneficial by being delivered to the actual parts of the plant that are growing. But then the other problem that results from that is that if you don't have downregulation, well, you will eventually have downregulation of carbon fixation. If you still have this same amount of radiant energy coming in, then you have an energy overload into the system and you have to invest in lots of protective mechanisms in order to survive a situation where you can't move that much energy into photoproductivity. So flow and function goes all the way back into the mesophyll and has all sorts of upstream effects on stress and carbon fixation. Okay, so obviously there are lots of places that HLB could be affecting transport. One would be at source loading. Another would be in the stem, right? In the transport tissues, and then another one could be at sink unloading. So the questions that we had were, where does the transport inhibition occur? How much does HLB actually impact the transport speeds, say? And then where are the transport bottlenecks? How does HLB affect photosynthesis? In other words, is there source sink attenuation, right? With the buildup of carbohydrates, do they downregulate photosynthesis? This is something that was actually assumed when we got started, and then we ended up with some surprising results. And then, of course, we ended up having to look first at how these processes work in uninfected trees to see kind of what the normal state of things is. So I'll start with that part, and then I'll work into HLB. Okay. So in terms of methodologies, I'll go over the two big ones that we use, and we use a number of other ones, but first is gas exchange on a couple of different scales. Some of what we do is, using like larger chambers to measure whole shoot photosynthesis. And then a lot of what we do is using ACI curves to kind of get at the regulation of photosynthesis. The basic idea of this method is that you expose the leaves to a wide range of environmental CO2 in the chamber, estimate the internal CO2 from that, and then the response of photosynthesis to internal CO2 allows you to calculate a couple of key processes, and that's maximum rate of carboxylation, that's this VC max, and RUBP regeneration, as well as triose phosphate utilization. I'm not actually going to show any results from these two. I'll just do VC max because everything that we've ever done in citrus, they all co-vary together, and so that if you talk about one, you're really talking about all three. The other big method that we use has to do with radioisotopes. We use carbon-14. And we have a couple of different methods. Some of them are non-destructive, so we use X-ray detectors and with very carefully controlled sets of conditions. This is a lead-covered X-ray detector, actually two of them, placed over the stem, and we can trace those pulses and measure the linear transport velocity. And then we use some destructive measures to look at allocation of that same labeled carbon after the fact. A couple of other methods that we'll come across. We have various methods of measuring starch, sucrose, and other non-structural carbohydrates. Measurement of bacterial titers, that's via quantitative PCR. There's some microscopy that you'll see. We have some metabolomics results and chlorophyll fluorescence. The bacterial titers, microscopy, and metabolomics, that's all just done through collaborations with people who have expertise in those areas. Non-structural carbohydrates and chlorophyll fluorescence is done in my lab. So when we look at healthy citrus trees, even just looking at the shape of the phloem, we could kind of hypothesize some differences from other trees. So this is just an example as a comparison. Typical reported sieve tube diameters for poplar is like 15 to 45 microns. And for citrus, it's 5 to 20. And if you use just try to model the relative flux, all other things being equal. So if you had two tubes like this, and then you had the same pressure gradient, the flux, that's Q, is related to radius to the 4th power. So if you look at those differences in diameters, you're talking about pretty big differences in relative flow, right? So roughly somewhere between 25 to 81 times different fluxes, all other things being equal, which of course we know they're not, but we can at least hypothesize that there's a lot of resistance to flow in a citrus tree relative to some more vigorous trees like poplar. And so When we get into actually measuring these fluxes, we see that that's pretty well reflected. So we have very slow transport rates. What Orlando did in this study is he just looked at the upward and downward flow of labeled sugars from a central leaf on the stem when there was new shoot growth present and when there wasn't. And we hypothesized that there would be more upward movement when the new shoot growth was present. There was a little bit, but the change wasn't so huge. We also expected there to be changes in speeds when you had, 2 sinks versus when you had only 1. And we didn't see that at all. So the translocation speed was, as far as we can tell, extremely stable. Not only that, but the distribution was almost entirely below anything that had been reported for an angiosperm species before that. So this orange line up here, that's our typical minimum angiosperm species. So that's the lowest that's been reported in any angiosperm. And the reported speeds in pines were somewhere in this range too. So citrus, very slow. So when we tried to look at sort of source sink attenuation in this context, what Orlando did is this illustration actually shows it being on the same tree, but he did it on different trees. He had vegetative trees that he did not defoliate at all, some that he removed 1/3 of the leaves, and this is alternating to go 1, 2, 3, remove the 3rd leaf and sort of throughout the canopy. He did one treatment where he removed 2/3 and one treatment where he removed everything except 1 leaf on every terminal branch. And that was roughly 83% of the leaves. We felt we ought to leave 1. Right? If for no other reason than just to be able to label. And one of the interesting things that he found was that there was no effect on transport speeds despite those really massive changes in source sink gradient, right? And he also didn't find any change in the leaf export rate, which we can measure a little bit more precisely. So, but he did find changes in the maximum rate of carboxylation. So the trees did respond by increasing photosynthetic activity. It wasn't a massive response, but it was definitely meaningful. And then he saw also differences in the allocation of that carbon once he was able to destructively sample these trees, right? So less allocation to fine roots when the trees were fully defoliated. Not too surprising because those trees respond by producing massive amounts of new shoots because they need the photosynthetic surface area to be able to continue growing, right? So big changes in allocation, changes in photosynthesis, but no change in transport speed, no change in leaf export rate. So Bilal in my lab followed that up by saying, okay, well, based on Orlando's work, we knew that there were changes in export rate in response to this presence or absence of the new shoot. One thing that's, it's not exactly unique about citrus, but it's different from the typical temperate zone tree, is that citrus grows in these spurts called flushes. And typically they'll have no new growth, no new shoot growth, and then suddenly they'll produce a bunch of new shoots. And those new shoots will grow, expand, and harden off in 3 to 4 weeks. So really, really high sink demand for a short period of time. And so we knew that leaf export rate did increase in response to that. So we said, okay, well, what about what can we do with that? Are they upregulating photosynthesis? And what he found was that so just to be clear, stage 2 is the just very beginning of new shoot growth, and stage 3 is just when they're almost at full expansion. And what he found was that the export rate only responded moderately, and the response depended on the position of the leaves on the mature shoot. And so the one that was actually furthest from the new sink is the one that responded the most. That's the blue line, is the basal leaf, right? So that's responding more. We're hypothesizing that that's because they're approximate to 2 types of sinks, right? Both the roots and the new shoots, whereas the apical leaves are only near the new shoots and sort of compete for export with the more basal leaves. And there was a pretty massive buildup of starch over the course of the quiescent period, right, where there's no new growth. And then that was just completely depleted during that new shoot growth. It's depleted in all the mature leaves. But then the response is that they don't actually upregulate photosynthesis at all. And in fact, photosynthetic activity actually declines over that period. So go figure. Not exactly what we hypothesized in terms of source sink attenuation of photosynthesis. And we're still working on, understanding that a little bit better. But so what we've learned from this is that one, transport doesn't accelerate easily. So nothing that we've done so far has measured an increase in linear transport speeds. Export from the leaf can increase under certain sets of conditions, but it doesn't always. Just because you have more sink demand relative to leaf area doesn't mean you'll have an increase in leaf export. Another thing is that we have a high degree of storage all along that pathway, leaves, and then also in shoots. And we expect that that's related to the stem providing a big buffer to the distribution of carbon. If you're more curious about that, I can talk about more evidence later, but that's the sort of our main source of like why is it not responding? Well, it's not responding because it has a lot of storage all along that pathway. And then we have these questions about the scale of the source sink attenuation signal, right? When we defoliate whole trees, we get a source sink response in terms of photosynthesis. But then when it's happening at the shoot level, then we don't see that response. Okay, so moving on to the next question of how does HLB affect photosynthesis? Now we'll get into how this disease is working. Initially, so Mark was a master's student in my lab, and he came to me after doing his first experiment of trying to compare photosynthesis between HLB affected and uninfected trees. He's like, yeah, I think it's not. I think it's not reducing photosynthesis at all. And I was like, no, you're wrong. So and so reported. And he's like, no, did you go back and read that paper, because I really think that they didn't, and he was right and I was wrong. But I still made him repeat the study. And he was still right. But it was, the results that we got were quite interesting. So this A here, that's net CO2 assimilation on a per area basis, right? And so you actually see an increase in the HLB affected tree relative to the uninfected tree on a surface area basis. And that's measured at the shoot level, by the way. It's not measured not just at the leaf level here. But another thing that changes is the total shoot surface area. So the total shoot surface area is dramatically decreased. And the result is that each shoot is photosynthesizing less, even though on a per area basis, it has a higher activity. So that suggests that carboxylation is not decreasing, at least not decreasing in the case of HLB. Dorcas is a PhD student in my lab right now. And this is a summary of a result from one of her studies where she was actually directly measuring BC max, and of course you see that there's like total overlap between those two, and so she's able to confirm that what he hypothesized from his work, which is that carboxylation is not really responding at all, at least not consistently. But stomatal conductance is responding. So you have a decrease in stomatal conductance, and you have a decrease in total surface area, and that's what leads to the resulting decrease in growth. We still have a lot of questions about well, why? Because it doesn't really make sense that you would just keep accumulating carbon endlessly and not reduce photosynthesis, especially something as costly as for nitrogen as Rubisco, but it is what it is. So the answer to that question of how does HLB affect photosynthesis is a little bit, but not in the ways that we thought, right? So it doesn't affect it in terms of internal foliar activity. It does affect it in terms of conductance and morphology. So then the question of how much effect does it have on transport? This is some work mostly led by Stacy and then Mirto in my lab. This is again using C14. We've got leaf export here on the right-hand side, translocation speed that's in the stem on the left-hand side. And you see they mostly agree that in one varietal combination, you see a decrease, pretty strong decrease in transport and export. And in another varietal combination, you don't see that much of a decrease. There's not any obvious differences in symptomology. So this wasn't a comparison between like a susceptible and a tolerant variety. It was just, we just happened to have two groups of plants and we measured them both. Yeah, so moderate decrease, not always consistent decrease in transportation. Then where are the bottlenecks? This is where, and where does the transport inhibition occur? This is where things really get interesting for us. So that same work by Stacy, one of the things that she did was quantify the deposition of callose in the tree. And there's a lot of controversy among people who study HLB about whether callose plugging in the sieve plates is primarily just a response to phloem dysfunction or whether that's actually the cause of phloem dysfunction. And one of the things that she did is so she measured the bacterial titer in stem segments, and in those same stem segments, the transport speed and the callose formation, the plug formation using kind of a microscopic method. And what she found was that there was an interaction between callose and bacterial titer and that the phloem translocation speed could be explained by those two factors. And that basically explained all of the differences that you saw between the two varieties. And what struck me about this is that it's at the local level. So within, a couple of inches of stem, you can predict the phloem transport speed if you know the bacterial titer in that segment and the callose deposition in that segment. And then Mark's work, same set of studies that I was mentioning before, he looked at starch concentration in the leaves and then also quantified the number of bacteria in the same leaf samples. And he found that at the leaf level, there was also a relationship, more bacteria means greater accumulation of starch. And there, in particular, there was basically an inflection point where you just got massive hyperaccumulation. Okay, so what that means is that wherever the bacterium is where the inhibition is occurring. And that's important because the bacterium is not evenly distributed. It is systemically distributed, but it's not evenly distributed throughout the plant. And so one of the things that happens is that you see the bacterium hyperaccumulates in the sinks. The fruit in particular are the best sink to look at because they're the sink for the longest period of time. Roots have a high turnover rate and new shoots, well, they become sources after a little while. But the fruit, citrus fruit develop over somewhere between 9 and 15 months, depending on the variety. So they're a sink for a very, very long time. And that direction of flow doesn't change, even though the sink sort of demand does change somewhat. This is looking at phloem in sieve tube. This is a sieve tube of the seed coat, which is the terminal sink. This is the end of the line for the phloem. And so just to make it clear, on the left-hand side, this is what a phloem sieve tube looks like normally under an SEM. And this is what a phloem sieve tube that's completely full of Liberibacter bacterial bodies looks like. If you look at the rest of the plant, you most of the time don't see any bacteria. The titers are so low that you have to look to be able to find the sieve tubes that have bacteria in them. Often when you find those tubes, you do see ones that have a large amount, but in general, they mostly just don't have any. But in the sieve tube, you see this sort of hyperaccumulation. So we thought that was a really good place to target our investigations about sink activity. So the hypothesis that we're getting from this is that essentially the bacterium is moving passively with mass flow, and that it's hyperaccumulating wherever there's a dead end of citrus or of phloem flux, right? But we're also then hypothesizing that that hyperaccumulation is responsible for the most dramatic effects of the bacterium. So one thing that Dorcas did, Dorcas's work is really going to change how we see the physiology of this disease. I'll just show a very small snippet, but she had this really cool setup because, so what you see here behind, this is Jean. Behind Jean, you can see like a little bit of netting. And some citrus growers, they've begun to grow citrus under like large acreage of netting that keeps out the vector. This is very costly, but with high value varieties, they can do that. So this is Murcott. Maybe the citrus that you're eating now might be Murcott. It looks like a Murcott, but what happened with these particular Murcotts is that Hurricane Irma came and it blew the netting off, and it took a couple of months for the grower to be able to recover his planting, and in that period of time, psyllids got in. They infected a bunch of the trees, and they provided the grower with a headache and me with a perfect natural experiment, because in the field, we don't get uninfected trees. And so we have a really hard time designing, field relevant experiments because there's just nothing to compare the infected trees with. But in this case, some of the trees got infected, a bunch of them did not. And this is a couple of years later, the infection is now thoroughly established, but they're not being infected among each other because he's eradicated the psyllid after covering the planting. So Dorcas had the opportunity to go and see trees growing in the field that with and without the disease. So she did a bunch of sourcing manipulation. I won't get into that part today. But one of the things that happened is she went out and identified based on symptoms which trees had the disease and which ones did not. After that, she went and tested, and it turned out that there was actually a group that were asymptomatic, but were still infected. So the original experimental design was 2 categories, infected and non-infected. And then the real experimental design was infected, symptomatic, and asymptomatic, and not infected. And it turned out that asymptomatic was really provided us with a really interesting perspective on what's going on here. So this is just fruit starch content and leaf starch content. And not surprisingly, the symptomatic trees, that's this peach color, they have more starch in their leaves. That's exactly what we expected. And the healthy have less starch in their leaves. And the asymptomatic do not have hyperaccumulation of starch in the leaves. Okay, that's interesting. But then when you look at the asymptomatic trees in terms of fruit starch content, so this would be a measure of like how effectively they're functioning as sinks, you see the opposite trend, right? So the HLB asymptomatic leaves are more like healthy leaves, but the fruits are more like infected fruits. So we're reading this as okay, well, definitely sink is the first thing that gets affected. That effect is earlier in the progression of disease symptomology than the foliar level symptoms. And that's consistent with research that was done a while back when HLB was new in Florida, where they observed root decline before they observed symptoms in the canopy. So we put this all together in we have sort of a model. So in terms of where carbon fluxes are affected, it's everywhere, right? But it's most dramatically at the sink level. We have changes in photosynthesis, but they're not due to internal regulation of carbon fixation. They're due to conductance and to morphology. And we have callose being deposited systematically throughout the tree, or not systematically, but systemically throughout the tree. And that affects just the total flux. But we have bacterial accumulation most dramatically at the sink level, and that affects the sink level flux more than the other fluxes. So to kind of test this in a field relevant way, Mariam has done 2 sets of experiments. On the right-hand side, this was earlier, she did a branch infusion with a couple of compounds, and on the left-hand side, this is later, she did a trunk injection of those same compounds. So what we have is an antibiotic, that's oxytetracycline, the same one that the growers are using. And then we have a callose inhibitor. It's named 3AB, and I don't remember the chemical name for what that stands for, but what we do know is it decreases the deposition of callose in the phloem and citrus. And then we have a combination of those two. And on the right-hand side, this is a C14 labeling study. In this case, she actually used sucrose in the lab on these shoots that had been sampled 2 weeks after doing the treatment. And what she saw was, well, the callose inhibitor primarily impacted import into the fruit. All three of those treatments increased the leaf export of carbon-14. And then if you look on the left-hand side, she injected in early April. It's a trunk injection the same way the growers do it, except we're using some slightly different compounds. And then followed the growth of individual fruits over that period of time, as well as the drop of the fruits, this is over the following 6 weeks, there's a natural physiological drop that occurs in May for us. In fruit trees, it's called June drop. We even sometimes call it June drop in citrus, even though it doesn't happen in June. So the fruit drop was diminished when she injected both the callose inhibitor and the antibiotic together, but not when she did just one or the other. It looks like maybe there's some change in the mean, so maybe they are having an effect in that direction. It's just not enough for us to detect. When she looked at relative fruit growth per day over that period of time, well, actually the antibiotic actually decreased the growth more immediately, but when she combined the antibiotic with the callose inhibitor, it actually increased growth. So we take this to mean that like we're kind of on the right path with sink activity being the primary suspect, but we have to actually decrease both the callose deposition and the bacterium to be able to see these effects. Okay, so from now we're going to transition into like trying to apply some of this knowledge. And so what we said is, okay, well, it's very difficult to deliver these molecules to some specific tissue in the tree, and that's not something that's scalable to growers. But what we can do is, knowing which sinks based on phenology, which sinks are most active at certain periods of time, we can time the injection of antibiotics to primarily impact one or another sink. And so if you inject 1 month prior to March, you should get an impact on that new root growth and just the very beginning of fruit growth. If you inject 1 month, inject in March, you'll get that. If you inject in April, you should get an impact on the initial part of the strongest demand in the fruits, as well as having an impact on the first vegetative flush, which should impact new leaf area. Similarly, if you inject in May, you'll get impacts maybe on the subsequent shoot and root development. And if you inject in June, you may impact the last part of the major fruit growth. So we did a preliminary study. We've expanded this to include all of the dates that I mentioned and a couple of others, but an initial study, the best we could do was 3 different times. And we did find that April injection increased both the yield and the quality. So that Brix acid ratio is kind of sweetness of the fruits. So these data would have been collected in February of this year, and the injection occurred in March, April, or May of 2024. So looks like maybe we're kind of on the right path in terms of being able to apply some of this knowledge to impact yields at least. We think that long-term effects on the trees might be better actually to target a different time, because if we can support root growth, we can combat a lot of the longer-term harmful effects of the disease. Okay, so that's kind of like the story in terms of trying to hunt down the effects of this disease on carbon fixation and transport. But my lab also does a fair amount with ecophysiology, and so we're trying to ameliorate the effects of this disease using some ecophysiological approaches. So when we look at how HLB affects the tree, you have, water is moving towards the canopy, transpiration is or is not occurring depending on how the tree feels that day, and that affects like leaf heating or cooling. And you have sugars moving towards the sinks, but in the case of HLB, you have less sugar flux, which leads to diminished root growth, which leads to diminished flux of water into the canopy, and you have water deficit symptoms occurring even when there's no water deficit in the soil. That exacerbates things like leaf heating. As an ecophysiologist, I always have to point out that the thing that drives all of this is not actually anything to do with the tree, it's sunlight. And so the major driver of transpiration as well as leaf level stress is that energy influx from the sun, which in a place like Florida is unrelenting. Got a little twirly sunlight. Okay, so what we hypothesized then was that we could change that by shading the plants. And so Anirban did this study shaded with different intensities. We used black shade cloth because we didn't want it to be an effect on light diffusivity or light quality or anything like that. We just wanted just quantity of light. And so the best we could do with that was these black shade nets. Shading these Hamlin trees, they were about 5 years old, fully infected, highly symptomatic when we began to shade them, and shaded them constantly for the following 3 years. So one of the things that we found was increases in photosynthesis, and Yuang and Jinhook, so they did the metabolomic side of things, and you see that the carbon fixation data was correlated with some of these carbon-related metabolites, not too surprising, and negatively correlated with pseudoheptulose, which is typically associated with like sink limitation. But one of the interesting things that we found was, and this is still like still trying to really fully explain this, is that we had increases in photosynthesis and we had decreases in foliar starch. So you have more carbon coming into the leaf. Presumably the plant still is not capable of moving carbohydrates more efficiently, and yet maybe it is because the carbon's going somewhere, right? But looking at some of this more physiology-oriented data, you'd hypothesize that there's a change in yields, and you do get that. Although the change in yields is not the same as the change in physiology. So with the 30%, that's that orange category, you get a really dramatic increase in yield. You get a less dramatic increase in canopy growth, and you get better growth and quote unquote happier leaves at the 50 and 70%, but you don't get more fruit. When I try to explain this to growers, I say this is like that, proverbial young person living in their parents' basement. They may be less stressed, but they're also less likely to reproduce. So, the 30% shade is the one who's, out and about in the world. It's a little more stressed, but it's also, more evolutionarily successful. In fruit trees, you can build a shade net, but you can also spray your shade on. So there's well-established technology called particle films, and these can be used. They're suspensions of solid particles in water. They're sprayed on the leaves. When the water dries, they form a film. That's hence the name particle films. They reflect light, and that's the main way that they function. But they do a lot of things by that light reflection. In terms of the physiology of the tree, you get redistribution of light, you get more shade on the outside of the tree, and you get a little bit of redistribution deeper into the canopy. So the leaves that are under high light might be more stressed. That stress is alleviated, and the leaves that are shaded and might not be that productive, well, their capacity to produce is enhanced. Another benefit that you get is that you get actually repellency of insects that are optically oriented. And so that includes psyllids. Don't only move by sight, but they do orient their flying when they're flying by sight. And this basically prevents them from identifying a citrus tree because citrus trees aren't usually white or pink. And so they just don't land with the same kind of frequency. Once they do land on the tree, they can find the new shoots, which is where they reproduce. So it's really just a matter of preventing them from landing as much as possible. We get somewhere between 60 and 90% efficacy from these treatments. But we also get a lot more growth. So this is the results of an establishment study. So we started these treatments in 2018 when we planted, or excuse me, in 2017 when we planted, and then followed them for the following 4 years. This is very initial. It's still very, very small fruit quantities. And the result is that applying the red or the white kaolin pretty dramatically increased growth, yields, all the things that we're looking for. And it did slow the rate of infection, although by the end of the second season, all the trees in the study were infected, but the higher growth rates were maintained by this redistribution of light. So what we're doing next with that, we continue to do some of the particle film work to try to optimize those treatments. And then also we're testing out different types of shade netting. So these are, I literally took these pictures Monday. And so one approach is to like build this shade. Another approach is to break up the landscape with some different non-host species and also provide shade. So that's what this study on the left is an intercropping study where we're growing citrus interspersed with other species that we think will be beneficial. Yeah, so with that, just want to summarize. HLB decreases carbon transport. That's occurring primarily at the sinks. Roots are a really important sink, and the effects on roots are the primary source of all of the ecophysiological interactions that HLB causes. But we can ameliorate that by a couple of different approaches to shading or redistribution of light within the canopy. Yeah, with that, I'd like to thank you for your time and invite any questions.
Really cool talk. Very excited to hear about physiology. I was really curious about your source sink manipulation part, where the defoliation changed the ratio of the source and sink, and that it didn't change export rates. I was wondering if you had a way or have a way to look at the duration of export instead of just the rate during a day, and if that changes, because there must be a diurnal cycle to when export's happening, and if that changes at all, or if it increases.
Yeah, that we do not have a good way to do that, and it's something that we have discussed. But so when we do a study like that, we're measuring it in like a greenhouse, and you can do it a couple of different ways with carbon-14. One is taking leaf punches, and it's a partially destructive sampling. Another way is to actually place an X-ray detector up against that leaf and read the decline in values. The problem with doing things that way is that the X-ray detector, if it's in any sort of like relevant outdoor type of environment, it's like that's way more radiation than is being supplied by the carbon-14 itself. So the only way to really do that method is in like a relatively dark environment, which then means that the whole plant is not really experiencing the kinds of conditions that we're interested in. So we have not measured changes in export in relation to time, but we don't. The short answer is we don't know.
The reason I asked is because I recently talked to Jessica Savage, who's working on phloem, in flowers, and she was thinking about it in terms of a mass balance approach with photosynthesis and just hole punches.
Yeah.
I was like, is that something you have heard about or?
Yeah.
Okay.
So that's been successfully done in a couple of contexts. And we actually are using mass balance approaches, but on a larger time scale than the question you're asking. So it only works for us if we're doing it like on a whole day scale again. So the benefit that we get from a mass balance approach is that we don't have to use carbon-14, and we can do that in the field, say. But there's not enough carbon gain in a citrus leaf to really see these, be able to measure these big fluxes. There's enough variability between the different areas that you would sample that it kind of overwhelms that signal.
Thank you.
That was cool to see. It was there, the bacteria is accumulating at the sinks. I remember seeing my parents' lemon tree covered with like sooty mold from an aphids or scale that were pissing out all the phloem and dropping it all over the leaves for the mold to grow. Do like scale if infected trees, HLB trees that have scale have lower, has anybody looked at like something that can suck out phloem and suck away the bacteria out of be an artificial sink? Like you just flood the guts of the sap sucking insects with the bacteria. And that might save the fruit.
I had not considered that.
IPM approach.
I'll go out and tell the growers to stop spraying for scale. No, we need them to leach the trees. Yeah, I mean, that I don't know. I would tend to assume that even though, they're pulling out significant amounts of sugars, it's mostly still along that pathway and not like
They're
not
not
there.
major sink. Yeah.
Yeah.
Scale is not a major sink.
So that use of antibiotics would seem to be a really good way to develop resistant bacteria. Are you seeing evidence of that? How long is this going to last?
So the short answer is we're not seeing any evidence of it yet. But even the growers are very conscious of this. And there are, they started out with restrictions on how many times they could reapply before they needed to stop for a while. But those were, they said 2 years on and 1 year off. But by the time they got to the end of 2 years and the growers were going, oh, but it's really helping. And then, now it's 3 years on and 1 year off. So like, I don't know. The short-term temptation of increasing growth that may outweigh the long-term risk calculation of developing resistance. So there's not evidence of it yet, but the assumption is that it's when and not if. And growers will tell you that too, right? They're not like, oh, that we don't need to worry about that, but it's just that there's so much urgency for them to increase growth and productivity that it's that out for them that outweighs the risk of developing resistance. But I don't see how it could not happen. You have one single, but the other side of that is that there's a lot of investment in developing new chemistries. And so unfortunately, those have not been super successful, even though a lot of things have been tried. But yeah, it's a major risk, and if we don't have new chemistries soon, then I don't see how we could not see the development of resistance.
Are there no antibiotics with different modes of action?
The only one that has been successful in citrus is oxytetracycline. They've also tried, they've labeled streptomycin, But actually streptomycin does not move very easily around plants. The explanation the chemists give to me is there's just a very large molecule with a lot of potential binding sites. And so it just grabs onto things as it moves through. And so it just doesn't really go around very much. If you put enough streptomycin into a tree, you will decrease the bacterial titer, but it's super inefficient. Those are the only two that have been approved. There are other chemistries that people say look promising, but I always wait to see some sort of like field-related evidence before I.
Go ahead, one more.
So I just want to follow up, for a lot of the treatment, you actually did chemical shit, whatever, you see beneficial, sometimes very unexpected effect, and you're trying to interpret based on transport for those. Have you looked at the potential direct or indirect impact on the bacterial proliferation? For example, the calcium inhibitor, that may not only affect the transport,
Right.
but also could induce immune response. Have you looked at the bacterial growth on that aspect?
We have not yet, but that's part of the experiments that are ongoing. So yeah, I mean, they could increase or decrease, bacterial reproduction. But yeah, so it's something we're looking at, but not something we have results on yet.
So a reminder tomorrow, 9 o'clock, Gavaza, if you want to come, some more to Christopher. Thank you for a nice